This is Breakthroughs, a podcast from Northwestern University Feinberg School of Medicine. I'm Erin Spain, Communications Director at Feinberg. The outbreak of a novel coronavirus in China is making headlines around the world. Here at Northwestern, microbiologist Karla Satchell is leading an effort to investigate the structure biology of the components of the virus to ultimately understand how to stop it from replicating in human cells through a medicine or vaccine. This work is being done with the Center for Structural Genomics and Infectious Diseases at Northwestern, which is funded by the National Institute for Allergy and Infectious Diseases. Dr. Satchell is the director of the center and a professor of microbiology, immunology here at Feinberg. Thank you so much for joining me today.
Thank you for having me here today.
Well, you have been in demand around here ever since news of this new coronavirus has come out. Tell me a little bit about the background of coronaviruses in general. This isn't the first time there's been a new coronavirus in humans.
So coronaviruses are simply a class of virus, just like flu is a class of virus, and they cause respiratory illnesses, and the virus can be spread. These viruses actually sometimes are very species specific, so maybe they spread only among bats, or they only spread among ducks or only among pigs, but these viruses can cause these dramatic diseases and can spread quite rapidly, and this is why they are a concern.
And that's what's happening right now. There's a lot of spread. There's been deaths. There's people that the numbers keep rising and rising. And that's why there's so much interest right now because we don't know a lot about this virus at the moment.
So that's what makes this of a deep concern is when you don't know the answers. So you need to have at least some data in some patients to analyze, well, how fast is it spreading and how is it spreading? So is this worse than SARS or is this really very similar to SARS, which was an outbreak in China in 2003, which caused a large number of deaths. But the number of deaths that we're talking about so far are nowhere on the scale, for example, of influenza. So we don't know if this is going to be a severe outbreak, and the concern is really that we don't know.
Again, you're a microbiologist. You spend your time in the labs. You're looking at these viruses under microscopes. Tell me a little bit, what does the coronavirus look like under a high-powered electron microscope?
So the coronavirus gets its name. Corona means crown, and it actually has a very crown-like appearance if you look at it under an electron microscope. It has what's called spike proteins, and these extend out, might look like the points on a crown, and that's where it gets its name.
So as you mentioned before, the coronavirus that infect animals, sometimes they can turn into new human coronaviruses, and that's what they're speculated has happened now. Is that right?
So what's come out from the evidence that's from what's called DNA sequencing, tells us what the actual order of nucleotides is on the RNA strand of this virus. And if you look at that code, you can compare it, well, what other viruses is it most closely to, has the most similar code. And surprisingly, that turns out to be bat SARS, which is a respiratory syndrome like virus that's actually circulating among bats. And this is in the same group that caused SARS in China 2003. But if it has a change in it that recognizes, instead of recognizing bat cells, it now has developed the ability to recognize human cells. And so now that's actually now when it gets into our lungs, it recognizes a human protein and has gained the ability, it would appear, to infect humans.
You're in high demand with journalists because your center here is on the front lines of responding to this particular virus as well as other viruses. Explain what the center is and what it does when such a public health emergency arises.
So the Center for Structural Genomics of Infectious Diseases was started in 2007, actually coming out of the original bioterrorism events. And what was recognized at the time was if you have an event happen, and that time the concern was a terrorism event, but subsequently we're more concerned about emerging infectious disease events such as the coronavirus that we're talking about today, is that if you have an event like this happen, you can't take time to pick up the phone, put out a call for applications. You can't wait for those applications to be reviewed. This process can take many months, even before people get started. What the National Institute of Health and the National Institute of Allergy and Infectious Disease would like is to have scientists in place already working who are trained all the equipment necessary already there, always working, that they can just activate on an instantaneous notice with nothing more than an email or a simple phone call. And this is what the Center for Structural Genomics Infectious Disease, this is our purpose. So while we sit around and wait for an infectious disease crisis, we actually do work on behalf of the entire microbiology and infectious diseases community, and not just viruses, but we're also working on fungi, on bacteria, as well as parasitic infections. We solve over 100 structures a year related to these infections, with hopes that the information that we generate will help our collaborators to develop new diagnostics, new drugs, or new vaccines. Over the lifetime of the center, we've solved over 1,200 structures. However, as I said, our main purpose is that we have all these talented people in place to address a problem should it arise. And this happened a few weeks ago with the novel coronavirus, where we were actually contacted by NIAID, asked if we were able to be responding. We have a full coronavirus group underneath the center. They're housed at Purdue, who were able to immediately respond. And after analyzing the depth of the problem, we actually decided to commit more resources to the center. Now every one of our 8 sites is responding in some way.
And that's important to note that while the center, you're the director of the center, we're here in Chicago at Feinberg, Purdue, all these other universities across the country have their experts who are part of this sort of consortium that contributes to the center.
So this brings a lot of expertise to the center. So as I said, I'm a bacteriologist, so we're actually a major bacterial component. We're working on antimicrobial resistance, as is our group at Argonne National Labs, funded by the University of Chicago and at Calgary. We have another group there, very dedicated to antimicrobial resistance, which is another major problem. But we also have very great viral groups at Purdue and Washington, St. Louis, who are actively responding to this upon request from NIAID.
So you said you get an email. Is there a quick phone call and you just start sort of assigning people on this? What does that look like?
The reference from Purdue, actually, they were on it. As soon as it came out in the news press, Purdue actually contacted me and said, how do I get the genome? I contacted the NIH and said, how do we get the genome? And they started looking, and we had that information within a couple of hours. The students in the laboratory down at Purdue started designing the constructs for the DNA that they needed to make proteins literally immediately, so that by the time the weekend was over, they were ready to go. They had their order placed, and they were ready to go. And then they switched to doing modeling studies and working on related viruses, things that they had going, in order to generate as much as they can while they wait for this first step, which is DNA synthesis. Simultaneously, our group at WashU, who's really involved in vaccine development, they began working with NIAID and have subsequently been asked to participate in projects as well. The rest of us got together and said, what we really do is do lots of structures rather than look at in-depth problem. We can do a lot very quickly. And so we decided to band together and put together a platform to look at 12 other proteins that we're going to be looking at as soon as our DNA synthesis is complete.
So where are we at right now in the process?
Right now, unfortunately, we are waiting. So the first step, unfortunately, a viral genome is something that shows up on your computer screen. And it is not actual DNA that you can put into a test tube. And so a very major advance, technological advance compared to any other epidemic to date, is the advance that's come from synthetic DNA technologies. So it used to be that you would need to actually get your hands on actual virus and actual DNA and use PCR to replicate that up and make multiple copies. Now, actually, all we have to do is ourselves sit down at a computer, type in the sequence that we want, and a company will use chemistry to actually synthesize that piece of DNA for us in the exact sequence that we requested. And so we've done this, and we're just waiting for that synthesis to occur. We anticipate our first DNAs will be arriving as we speak, hopefully today, perhaps tomorrow, and we expect another batch to begin to arriving next week.
And again, in order to investigate the proteins in the virus and look for the drug or vaccine that may stop it from replicating, you're creating these 3D models of the structures that can be analyzed, you're saying, like, really with like graphic software. Again, tell me, how does that work from the whole process, from the time you're cloning it right now, from what's happening right now to the finished product?
So we'll get the DNA and it'll come fully cloned so that we can actually put that now into bacteria. We use the bacterium E. coli, and E. coli will become basically a protein factory for us. So it will start to replicate and make that protein at a very high rate using the code on the DNA to identify what order the protein amino acid should be in. We then take those E. coli and we break them open. We release that protein and purify it using chromatography columns. And then we use that protein and we make crystallization conditions for it that allow that protein to pack into a very dense crystal that is a solid repeating pattern. And we see these crystals, these microcrystals will grow in a dish, and then we can take those to Argonne National Labs where they have the advanced photon source that we can actually take these crystals and blast them with X-ray beams. And that will lead to what we call a diffraction pattern. And that pattern can be used using computation to identify what the three-dimensional structure of the protein was. So it's a way to get a three-dimensional picture, or, if you will, of what that actual structure was. So we can take a picture using a camera of our face. The X-ray beams and the diffraction patterns can do it of a very, very small protein or protein crystal.
And these 3D visualizations are pretty cool. They look like tangled bunches of curly, colorful ribbons. Is that how you would describe them?
So that's the depiction that we use for them. So if you think about if you have a strand with lots of hairs coming out of it, right? If you had just a ribbon with lots of hairs coming out and you just stripped it of all the hairs, it would actually look like a really nice ribbon. But in fact, for all of those ribbons, there's a lot of chemical bonds that are extending from that. And it leads to actually more of a blob-looking structure. But if we want to show that in a way that's very tractable, that we can use and do comparisons, it's easy to strip away a lot of that information just to what we call the backbone. And that's the ribbon presentation that you frequently see. And it does look like a balled-up Christmas ribbon. However, it's not a balled-up Christmas ribbon. It's very defined folds in an exact pattern mediated by chemical bonds that allow that protein to interact either with other proteins, to interact with chemistry or chemicals. And so in our case, we're really interested in how these are going to bind and interact with drugs.
The proteins of this virus, some are very similar to the previously identified SARS virus, but that doesn't mean that any medications that might work for SARS will necessarily work for this virus.
That is correct. So if you think about how chemical bonds interact, it's one chemical interacting with another chemical. If you have that protein changes just ever so slightly, so one of those bonds is disrupted, you can change how that drug is going to be able to bind to that protein. If you change multiple of them or change even a very large piece of it, then you're going to cause a dramatic change. And so what we're seeing in the coronaviruses is that they're about 70, even up to 99% identical. But if you have 30% change, that may be enough to actually make current drugs for SARS completely ineffective. Or what we're really hoping is that those changes are not so dramatic with regards to the drug interaction and that those drugs are going to work very well. We do know already that the changes that have gone on in this spike protein probably are sufficient, and that if you have prior exposure and you've had SARS before, you're not going to have antibodies and you're not going to be protected against the coronavirus.
You just walked us through sort of the process and how quickly everyone is able to mobilize and respond to this, but the fact that you're able to do this so quickly really has to do with advances in technology and sort of a lot of groundwork that's already been laid to do this. Tell me about how things have changed in recent decades to speed up this process.
So in 2010 was when next generation DNA sequencing technologies came on board. And we were able to sequence DNA before that, even going back well into the early 1990s. But the speed at which to do it, and to do it in very high density and from not particularly good material, has really come about since 2010. An example would be if your crime story, right, their ability to identify DNA at a crime scene or DNA from bones that they're discovering in graveyards, that's really getting this high density information. And so one of the things that's going to happen in this epidemic is they were able to take clinical samples. They were suspicious they had something new. They were able to get sequence information on that, identify it as a coronavirus, and more importantly, identify it as a new coronavirus, probably certainly within a day or two, and possibly even within hours, a process that in the past might have taken a month even on itself. The Chinese released that information to the global research community, who then began working. We're already seeing now publications come out about how this virus interacts with receptors, how does it recognize humans. There's beginning to be development already for vaccines and drugs and diagnostics. The epidemic is documented as having started December 29th. Studies out today suggested possibly back as far as December 17th. But if you think about it, that's really only 30 to 40 days past, and we already have a density of information. That is an unprecedented speed that has occurred.
And so while we're able to sort of gather all this information very quickly and have a lot of people working on it, what are the chances of having a drug or vaccine in time to help people who are affected by this current outbreak?
There's a delay involved in developing any kind of a medical intervention because we do need to be concerned about safety. But trials are already being started right now in China to determine if any drugs that we know treat viruses can actually be used and applied if people will get healthy on this. So that's the first drug that you can do. But if you have to develop something new, then you actually have to get to the research lab. You have to go through phase 1 and 3 trials. But usually what they'll do is dramatically expedite these events. A vaccine can be developed in a heat kill and production can be ramped up. But we are looking certainly on the scale of months. And maybe that won't be in time to handle this particular initial spread. But as that virus gets out into the community and becomes a disease that simply exists, it will certainly be able to be treated then.
So you also have your day job here at Northwestern and microbiology immunology here at Feinberg. Tell us about why did you want to take on this role as center director? You have your own lab. Tell us what you're working on in your lab and how does that fit in with your duties as the director of the center?
About 3 years ago, I just decided I was looking for a new leadership opportunity. And when I heard that Wayne Anderson was going to be stepping down, I realized it would be a great opportunity for me. And the reason is because before I was the director of the center, I was a community requester of the center. So I was using the Center for Structural Genomics to do structural biology that I needed done for my research program. And this was done in collaboration with the then director, Wayne Anderson. The program that I'm really interested in is thinking about the diseases of cholera and the diseases of Vibrio vulnificus, which causes a very nasty necrotizing fasciitis, either from swimming in the sea or severe sepsis from consuming raw oysters. And both of these bacteria produce toxins, and these toxins are extremely large proteins that get into our cells and manipulate those cells in the cell biology. In some ways, they have a function almost kind of like a virus, except it's just the protein itself that gets in rather than DNA that gets made into a protein. And once it gets in, it manipulates all kinds of intracellular processes. And we're really interested in how those work. And most recently, we identified in a paper that just came out January 14th that shows that this toxin is required during cholera to completely block the ability of our host immune system to see that we're infected with cholera. And so that was a very exciting finding. It's been known that cholera was doing something sneaky all along, but it was never realized that it was related to this particular toxin.
Now, where was that published?
That was published in Science Signaling.
All right, we'll put a link to that on our page so people can check it out. And the part about the sepsis in oysters, how widespread is that?
So here in the United States, there's around 200 cases a year, and that causes 30 to 40 deaths per year. These are particularly rapid deaths, can occur within 48 hours of having consumed the oysters. The death rates are quite high. About 50% of cases from oral consumption are lethal. About 25% of wound infections are lethal. So, and the ability for it to cause this disease, in particular its ability to move from on your skin or in your gut to causing sepsis, is related to the toxin that we study. We're trying to understand how this toxin helps the bacteria move across those barriers. And the disease is rising now with climate change. So the bacteria like to live in warmer waters. Of course, if you have warmer waters and you have that over a greater period of time or a greater geography, that's causing increase in the number of cases, which is why we're concerned to learn as much as we can about this particular disease.
Well, we are going to have you back on to talk more about that topic another time. I know you're very busy with this coronavirus right now, so we will let you go and continue your good work that you're doing there.
Well, thank you so much for having me here today.
Did you know episodes of Breakthroughs are accredited for CME, Continuing Medical Education for Medical Professionals? Go to our website, feinberg.northwestern.edu, and search CME to claim your credit.